Yeah, and you guys can be in front of the screen. Yeah.
So our project is Super Screens and our team.
I'm Selena.
I'm Pujit.
I'm Jackson.
I'm Ramya.
And Yana should be online.
Yeah,
Mhm.
here I'm.
Oh, I think it might be on a
Go back
different,
to
yeah.
the tab. So. You just press there. to the right.
Okay, so first with our motivation. So we were looking at a paper that Manu suggested that we look at, and the paper basically talked about this study in which school children like source broth recipes from their family for different soups. And researchers tested 56 of these broths, and they found that 5 of these broths provided like 50% in vitro growth against people's malaria parasite, and also that 2 broths had similar inhibition levels to dihydroartemisinin, which is a leading anti-malarial. So basically this kind of showed that the anti-malarial activity in soup broths and like more generally in like food items. And another thing to like take note on is that drugs for neglected tropical diseases are very lacking. According to this like article in Nature that was published in 2018, the number of deaths and disability adjusted life years, especially in like countries that have like low and middle income communities, is very high. And so it's important to think about ways we can create drugs that are more accessible and frugal. And also drug screening in general is really challenging. So like for general drugs, we already have like insufficient funding and like poorly understood drug discovery paths. But for neglected tropical diseases, the challenge is even more amplified because of like increased like paying attention to costs and safety requirements. So this is like a big problem that was discussed in this paper. And like a few quotes from that are like, currently available drugs are inadequate to these diseases, and they inflict a heavy economic burden, especially on LMIC economies. And overall, there's an urgent need for new, safe, low cost, and efficacious treatments of drugs. So we thought, like, why not just screen approved and traditional drugs that are approved for other purposes or anti-pathogenic activity, because then we wouldn't have to go through this whole like new drug vetting process. So our whole goal for this project was developing this like frugal like drug screening platform based on currently like approved medicines.
So here are the 4 most important functional requirements for our project. The first one is that it's high throughput, so we can go through a lot of drugs and different pathogens. It needs to be versatile, so we can screen a wide range of drugs and pathogens. It should also be scalable so that many different people can use it in different settings, and it has to be robust, so reasonably accurate when it comes to screening activity.
Yeah, so I'll be explaining you methods. So here are some conventional methods like MIC, MBC, and disc diffusion. So in MIC, it stands for minimal inhibitory concentration, where we will see the antimicrobial activity with respect to the bacteria. We will see the growth in the 96 well plate. So yeah, so this is the composition, and actually we will take 2 controls in this. So first we will have the antimicrobial control, and then we will have the control of the bacteria which we are using or which we are willing to test, and then the concentration will get reducing like 16, 8, 4, 2, like that it will get reduced. And whenever we will see the minimum activity of the bacteria, then we can say that that concentration can be the suitable one. So that is the MIC. The MBC is minimum bactericidal concentration. In this test, we will see the bactericidal effect, like how the bacteria dies in the with respect to the antimicrobial which we are testing. So yeah, so in herbal testings, this is pretty effective and cheap. Like we will perform it. We can perform it handy. So yeah, so and then the disc diffusion. So yeah, so here this is the conventional method. Just a so this is a conventional method. So here we can see the plaques where the we can see, like in the image, the Petri plate is there, and around the antimicrobial substance, which is of known concentration. So we know how much antimicrobial substance which we are using, and the plaques are there. So we can tell that there is no growth of bacteria. So this is the visual thing which we can see actually. In the plate we can actually measure the concentration, but here we can see it is giving the bactericidal effect. So yeah, the bigger plaque, the more efficacy of that bacteria is there. So I'll leave you with a comment, like if a very interesting article I read about that many astronauts go in this space. So they also get some cold there. They might get some cold, fever, anything. So yeah, so in there, if you are thinking like they'll get the same medicine, like if we are using some Dolo 650 here, so that 650 mg if we are using, and if that is the same concentration they are using, it won't work there. Actually, they'll need something about 20x times because there the microbial concentration is different, all the conditions are different. So yeah, it's not with the body to body, but it's with surrounding also. So yeah, you can take from it.
All right, thanks, Yadnesh. So those are the conventional methods for an experiment like this. Now we're going to briefly detail our method. So firstly, we would prepare a drug library from readily sourced medications, supplements, and even culinary ingredients so that we can test a wide variety of different kinds of substances for antimicrobial properties. We would culture pathogen samples in 96 well plates and apply live dead stains to these samples. Then we would add dilution of the different substances from our drug library to these samples. We would use the squid to image these samples, and we would process these images to determine whether the pathogens survive in the presence of these kinds of ingredients from the drug library. And ideally, we would look for a way to automate the process. So the way that we're envisioning right now is through 3 machines, and we're going to detail each of those machines for you right now. So the first is Opentron. This is an automated pipetting machine. So this is what would be used in terms of, getting everything into the 96 well plate, basically making sure that the pathogens can be contaminated with the diluted ingredients so that we can test the different kinds of reactions with ingredients and see which have antimicrobial properties. The second is this automated Dorna robotic arm. And I think rather than explain what this does, it grabs the well plate. We could see a video of it right here. I'll let that play for a moment.
This is someone else's video, not our video.
It is our video because this is at Jordy's place. You can see the squid right there. It's an implementation of this. This was done in Sweden. I'm assuming you guys are very close to getting here.
We're still waiting for the servo to come.
Yeah, that's okay.
And of course, the final of 3 machines is one that we're all familiar with by virtue of being in this class, the squid. And this squid is used to image the 96 well plates that we were discussing.
Yeah, so now let's move on to the progress we've made. So first we tried imaging with the squid. We got the computer to talk to the squid. Second, we tried controlling the robotic arm so that it moves to the positions we specify.
This is our video.
Yes, this is right next door. So basically we can program the positions we want it to go to, and it will go there at the right time that we program it to go to. Yeah. Here, we tried culturing soil bacteria so that we can use it as a test. We dug up some soil from the Shriram Sankantiru. And I did that. We dissolved the soil in some water and then dripped the water onto some agar place to culture our bacteria. We made some streaks, they overgrew. We also made some suspensions in LB broth, which can be easily pipetted into the 96 well plates. We also ordered a live dead stain. So the way it works is that when a cell dies, the cell wall gets permeated by the red stain so that it turns red. But when it's alive, it's green. It hasn't arrived yet, so we couldn't test it, but it should arrive today. Because we were waiting for the stain to come, we tried to borrow some stains from other labs. Unfortunately, the lab next door, they usually study mammalian cells, so we only had a stain for mammalian cells, which is this blue thing here. It's called trypan blue. We tried looking at it under a microscope. However, it didn't show a very clear and obvious difference between dead and alive. It also required looking at it on a glass plate. So we thought we could just wait for our live dead stain to come so we can just determine live to dead ratio based on fluorescence.
And then we also created an initial drug library list. So we have like 25 plus drugs-ish, but they go into 5 main categories. We have pharmaceuticals, food substances, herbal medicines, Ayurvedic, and Chinese medicinal herbs. And here are like some of the things we were thinking of testing. So I think we thought it was important for us to test pharmaceutical drugs as well, just to have like controls. So we see like how like pharmaceutical drugs perform versus like food substances versus all these other herbs.
I like the fact that kimchi is there.
Well,
I know I feel good.
so now we'll talk about our future steps. So
Yeah.
we made some progress, but there's a lot more work to do for this project. Our ultimate goal is to build an automated drug screening system. That constitutes a system that can run drug screening experiments to measure potency, as we discussed. And some of the work that we still need to do is measure potency through like how drugs slow the pathogen rate, which we can ideally do without live dead stains, but that requires more precision imaging. We also, like as Selena described, hope to use the live dead assay to measure potency by comparing live to dead cells. We need to standardize drug concentrations. This requires like measuring effects across the various drugs to see at which levels things happen, as well as standardize it across the various substances we do have in our library. And we also want to compare the effects of different concentrations as described. So right now we're testing on soil bacteria, just to see as like a proof of concept how the system would work. Ideally, we would want to test on a real pathogen, such as malaria parasite or others. So the automatic screening pipeline is also a robotics problem and like a mechanical problem too, because there's various devices, the Opentron, the squid, the robotic arm, that we want to connect in an integrated way. And as Jackson described, so the ultimate goal is to show that such a system could work, and then try to deconstruct it and make it as simple as possible, and as also as like cost effective as possible once such a pipeline can be shown to work. So that is the ultimate goal of the project. And yeah, thank you. That it? Yeah, thank you all, and thank you to Hope, Hamshan, Ian, Ellie, Dina, and Manu. Yeah.
You guys are the first team that is on time. That is fantastic. We actually have time for 2 questions and then we'll jump. So I mean, first of all, this is fantastic. And, the many people online might not have realized that all the 3 instruments that you guys shared are all co-located now. You have them. I mean, one comment I wanted to make, something that you guys should just do immediately. Squid is a very powerful imaging system. If you guys remember some of our TB data, we can just see bacteria in phase contrasts. So even if you don't have any live dead stains yet, we can directly just look at growth rates. Just image the bacteria that you discovered from the soil and watch them grow. And then you would have a control growth rate, and you would have a growth rate in a context of a medicine or a drug. And the rate of growth itself can actually be used to identify many things to begin with. And, I think there is already a pipeline that we have built for identifying bacteria, counting them. So there is a lot to be done just purely with what you have working. You have bacteria and you have squid. Put those broths in and actually just start imaging. So you get just also certain set of dynamics associated with it. We have time for another question. Any comments, questions before we jump to the last talk?
Yeah, I will say that like we tried to do that a little bit. We tried actually putting the bacteria on like a plate and imaging it. Maybe it was just our inexperience with using the squid, but we were unable to like actually like visualize things. So that was like the first step here of like,
Yeah, I think I'll show you guys tomorrow right after the class. Let's do some experiments. Yeah.
yeah, thanks.
I think we were using a very low objective too. It was a 10x, so maybe it would be beneficial. to
Yeah. No, we would switch to a 40x or a 60x for this. Yeah.
Try the 40x, right?
I don't think we got to
Yeah,
get the right distance.
we
Yeah.
tried, but the distance between the plate and the objective was like not correct, and our adjustments just didn't work. But
Yeah. Simple tricks. Yeah. Okay, any other comment? I think one thing that I'm just very excited about is also your drug library. So keep working on that to actually put it together, because it would be really fun to have that as an open library, make many copies of it and share them with people too. So we have a version of this set up in Sweden. We can imagine setting one up in Sudan, sharing the libraries for different pathogens, because pathogens cannot be shared. Drug libraries can be shared.